Welcome to today's George Khoury Memorial Lecture. A few brief words about George. George is a graduate of Princeton University and Harvard Medical School. He came to the NIH in 1976 as head of the virus tumor biology section in the Laboratory of Molecular Virology, the NCI. In 1980, he became chief of that laboratory. The following year, in 1981, he received the Arthur S. Fleming Award for Outstanding Government Service. And in 1987, the year of his untimely passing at age 43, he was elected member of the National Academy of Sciences. During his short career, George published over 140 papers deciphering mechanisms of eukaryotic transcription. He had a really stellar group of trainees and collaborators. Many of the individuals on this list will be familiar to today's audience. And this lecture has been given by a similarly stellar group of investigators going back to 1995 with Phil Sharp and continuing until the present, almost uninterrupted. And today's speaker will recognize the picture of Pat Moore over here on the right. Pat was the 2017 Khoury lecturer. So it's really a pleasure to welcome Dr. Ewan Chang for today's Khoury lecture. Ewan received her undergraduate degree in biology from Stanford University, an MD from University of Utah School of Medicine. She was a resident in anatomic pathology at UCSF and a fellow in neuropathology at Stanford University Medical Center. In 1993, she took a position as assistant professor of pathology at Columbia University. She worked her way up through the ranks at Columbia University to full professor. And then in 2002, she became professor of pathology at the University of Pittsburgh School of Medicine. And she's currently Distinguished Professor of Pathology and American Cancer Society Research Professor at the University of Pittsburgh School of Medicine. Ewan has received a large number of honors and awards, far too many to list here, but just a few. She's a recipient of the Robert Koch Prize, the Paul Ehrlich and Ludwig Dartstetter Prize. She's elected fellow of the American Academy of Microbiology, the National Academy of Sciences, an elected member of the Association of American Physicians. For many years, Ewan has been a pioneer in the discovery and characterization of human DNA tumor viruses. Early in her career, she was interested in using representational difference analysis to study the genetic origin of brain tumors. She later applied this technology to Kaposi sarcoma tissue, resulting in the 1994 discovery, together with Pat Moore, of Kaposi sarcoma-associated herpesvirus, or KSHV. As many of you know, Kaposi sarcoma is a highly prevalent cancer in AIDS patients. She later used similar technologies to discover Merkel cell polyomavirus, the causative agent of Merkel cell carcinoma. So Ewan not only co-discovered these viruses, but has also been a leader in characterizing their basic biology, how they replicate, and how they induce disease. So it's really a pleasure to welcome Dr. Ewan Chang for today's Khoury lecture, Human Oncogenic Viruses, Nature, Discovery, and Running Around in Circles.
Eric, thank you for this introduction. It's particularly a pleasure to be here for the George Khoury lectureship because over the past 15 years, in terms of our work with Merkel cell carcinoma, it's really been greatly informed by the foundational work that George has made in the field of SV40 and other related polyomaviruses. In this talk, I'd like to tell you about the work that our lab has been engaged in on cancer viruses and cancer virus discovery and some general principles that we've learned from this process. Okay, so viruses can cause really explosive symptoms when they're actively replicating in the lytic mode. And these are some examples of viruses that have had a really high profile when they are in that mode. However, after initial infection, there are many viruses that go into a stealth mode or latency, where they really severely curtailed their expression of viral DNAs, RNAs, and proteins. And that's to keep under the radar of the really amazing immune surveillance system that we have. So most of the time, these less ostentatious viruses, they don't get a lot of press, but we are beginning to understand them, some of the things they do, They're part of the native flora that we have on us. They cover us, they reside in cells in us, they circulate through cells throughout our whole bodies. And so under some special circumstances and or co-risk factors, some of these viruses can cause cancers. Now, hundreds of viral species are known to infect humans, and yet 7 of these viruses cause cancers, which I've listed here. And despite the small number of these viruses that are directly involved in human malignancies, global studies show that 1 in 6 cancers worldwide have a viral etiology. So although this estimated attributable fraction is significant, it's probably substantially less than the actual fact of how many cancers are associated with viruses. This is mostly due to the fact that developing countries are particularly hard hit by viral cancers, individuals in developing countries. And, they frequently have almost non-existent cancer registries to keep track of these processes. This is a pie chart from the WHO Global Can, which shows the distribution of tumor types worldwide. And in developing countries, we can see that the leading causes of cancer are lung, breast, prostate, colon, colorectal. But in contrast, if you look at the Kampala Cancer Registry, for example, things are very, very different. The viral associated cancers are very, very common. You see here that cervical cancer, KS, these are cancers that are associated with viral infections, comprise up to 50% of cancers, both in women as well in men. And so statistics like these make virus research and the search for new pathogens really intimately connected to cancer research. Now, humans are known to be infected with more than 200 species of viruses. And the first discovery or identification of the human virus was the yellow fever virus. And this was by 4 Army doctors led by Walter Reed at the very beginning of the 20th century here. They used filtration isolation techniques and this time-honored method of self-inoculation with either diseased tissue or the vector to prove causality. And unfortunately, one of these Army doctors, Jesse Lazier, died from this type of experiment. But from these inauspicious but really heroic beginnings, one can see that the discovery curve for human viruses really goes up, but there's seemed to be an inflection point halfway through the 20th century here. Prior to this, viruses were identified by filtration or tissue culture, but with molecular biology originating in the 1930s and the 1940s, this rate of identifying human viruses really picked up momentum in the 1950s and 1960s. And it's associated technology of this molecular biology explosion really was related to this increased risk, I think. And so subsequent to the onset of molecular biology, each of the 7 human cancer viruses have subsequently been identified. So with the advent of molecular biology techniques, DNA-based detection of tumor viruses became possible. And so I'd like to just share with you the case of Kaposi sarcoma, which is very illustrative. This is KS, the tumor. The clinical presentation are purple violaceous spots on the skin. if one were to take a biopsy of one of these lesions and to look under the microscope, one would see this subepidermal proliferation of cells, which on even higher power shows spindled appearance, lining very irregular vascular spaces. Here's red blood cells in these irregular vascular spaces. And this is an endothelial cell tumor. It was, of course, described first in 1872 by a pathologist named Moritz Kaposi, but it really wasn't until the 1980s when the clinical presentation of this tumor took a really different, a real dramatic change from what it was before. It had always been an enigma. You know, it was seen very rarely in European countries, but occurred very commonly in African countries. It affected young children in African countries and was frequently fatal, but it was an indolent disease among elderly men in North America and Europe. So this changed with the onset of the AIDS epidemic, and this is clearly a harbinger, actually, of the AIDS epidemic seen here. And so this is really an amazing graph. And what I think is really remarkable about this is that you can see this is Sears data from San Francisco County or city between 1975 and 2014. And you can see this rapid increase in rate of KS in men between the ages of 20 and 54 in this study. And this rapid increase in rate was not seen in colon and rectal cancers, not in lung cancers, although there is a little increase in lymphoma, nothing like what is seen with this peak in KS rates. And this occurred between 1980 to the late 1990s. And this was a time when actually, personally, I was in medical school. I was doing my residency at the San Francisco General at UCSF. And every autopsy I did during that time seemed to be an AIDS autopsy. And we saw so many cases, so much KS, not only on the skin, but disseminated throughout the internal organs of these individuals that would come to autopsy. So clearly this was new. It was a different profile that we'd never seen before. In medical school, in our textbooks, we learned that cancer didn't occur as an epidemic. But what we see here is that we do need to consider cancer as an infectious disease. So to look for an agent, a lot of really amazing epidemiology was done at this time, which really suggested that there was an infectious agent. And so to look for that infectious agent, we took on this technique called representational difference analysis. And what it basically does is to subtract by hybridization DNA from a disease lesion, in this case, Kaposi sarcoma, against normal tissue from the same individual. And through iterative rounds of this subtractive hybridization and PCR enrichment, what we found by the end of this process were two specific bands that actually, in the end, comprised less than 1% of the entire genome of this new herpesvirus that we found in Kaposi sarcoma. And even with that small amount of DNA, both our lab as well as others who were working in the field were able to make disease associations. So this new human herpes virus was not only associated with Kaposi sarcoma, but with two lymphoproliferative disorders. One is multicentric Castleman's disease, the other one is primary effusion lymphoma. From these lymphomas, we were able to establish cell lines. From these cell lines, we were able to sequence the entire genome, clone and sequence the entire genome of KSHV, make a phylogenetic characterization. It is a gamma herpesvirus that is very much like a sister virus, Epstein-Barr virus. And here's the genome sequence. And, we found so many things in terms of, what the virus encodes in order to affect a transformed phenotype. From the cell lines, diagnostic assays were developed, and this allowed us to look at the natural biology of the infection in different populations. This is a slide that I want to show that sort of summarizes what we learned from this foray. And this is a meta-analysis of HIV/AIDS in transplant patients. And it's this is a standardized incidence ratio registered, I should point out, on a log scale. And this is from Andrew Grulich's group that was published in The Lancet in 2007. And what it shows is that among a wide variety of cancers that are associated with infection, what we see is that AIDS and transplant patients are at a much higher risk, above 1 here, at a much higher risk for these tumors. So from our study of KS, and here is Kaposi sarcoma, the other name is human herpesvirus 8. And this is a 20,000-fold increased risk for developing KS in HIV/AIDS patients. And so what we can do from this information is that we can use this type of association to look for other cancers that might have a virus. So this is a paper that was published in The Lancet from Eric Engel's group, who's at the NIH, and it describes the fact that Merkel cell carcinoma was found to be at a 13.5-fold increased rate in immunosuppressed individuals, in HIV positive individuals. And so that was a really intriguing study to us because of that clinical profile of association with immune suppression. And so what is Merkel cell carcinoma? It's actually a pretty uncommon cancer, but it's a very aggressive skin cancer, probably much more aggressive than melanoma, and 50% of advanced cases survive less than 7 months. The risk factors, again, not only is immune suppression, not only in AIDS patients, but in transplant patients and elderly individuals. And we know another risk factor, which is sun exposure. And so because this tumor demonstrated such a strong association with immunosuppression, my lab thought that this was really an ideal disease, an ideal candidate to look for a new tumor virus. And so to look for a new pathogen, we applied a methodology that we named digital transcriptome subtraction, which took advantage of the tremendous advances in sequencing technology since we applied RDA in 1993 to KS lesions. And so while KS, in our experience before, was identified by RDA, which is a DNA-based methodology, it's a methodology that really greatly favors large DNA genome viruses. It requires normal matched tissue as well as sterile tissue. And so with DTS, we go under the assumption that a pathogen acting as a direct carcinogen would produce viral RNAs in each and every tumor cell, and that these viral sequences could be detected by deep sequencing. And so this work was performed by Hui-Chen Feng, a really talented postdoc in our lab at that time. And so instead of physically subtracting the DNA from a tumor, comparing it to healthy tissue in a microfuge tube, what Feng did was to sequence the entire MCC transcriptome. and then use in silico subtraction to subtract out known human sequences in the deposited national databases. And so this was timely because the human genome had just been sequenced in 2003, and the cost of sequence had really dropped dramatically. And so after isolating RNA, making a cDNA library, and sequencing, what Feng did was to make a file of high fidelity sequences. These were sequences that were just right, that we really had high confidence in. And then silico subtraction was against RNA databases, genome databases, mitochondrial diseases, mitochondrial databases, and immunoglobulin databases. And when we did this subtraction, what came out were about 2,000 sequences that we considered were non-human. And this comprised about 0.6% of the original HiFi sequences. Then we decreased or made less stringent the filter and compared these sequences against viral databases. And we were able to find one cDNA that was similar but not identical to the T antigen that came from an African green monkey. And this was a polyomavirus. So it was really actually pretty exciting at this point. And within a week, Feng was able to walk the genome and to figure out that this was a 5.4 kilobase virus, a small double-stranded DNA virus. And the genome is really actually quite simple. Of the 5.3 kb sequence, the genome is roughly divided almost evenly into two bidirectional transcriptional units. There's an early region that yields 3 tumor antigens or T antigens, large T here, seen here, small t, and a T antigen called 57 KT. Subsequently, Denise Galloway's lab has identified yet another protein product called ALTO for alternate LT open reading frame. And then in the late region, what we have are structural capsid proteins, VP1 and VP2. And again, subsequently, a microRNA locus was found in the early region. These two transcriptional units are separated by a non-coding control region that contains an origin of replication. Again, very much like SV40, the virus that George Khoury had worked on and really made some foundational studies. So we were able to incorporate a lot of information of all the work that had been done. We found that the MCV, the virus, was clonally integrated in 80% of Merkel cell carcinoma tumors. And let me just show you, bear with me, with the transcript organization that is linearized in this diagram. And here we can see the small t, and these protein products of MCV makes use of every open reading frame. The small t reads into an intron that is spliced out by the large T, and it splices into frame 3. So 57 KT and ALTO, we know their splice pattern. We know when they're produced, but we don't know much about their function right now. So I'm going to concentrate more on the small t and the large T. Subsequent to the finding of the virus, many groups have identified very interesting binding partners, interaction partners to each of these viral oncoproteins. But when you look at the structure of these T antigens, in particular the large T, you can see that the virus replication functions. The large T is the spliced protein that is the major replication protein. And that has the replication function of having a helicase ATPase domain and an origin binding domain in the C terminus. When we look at all of the Merkel cell carcinomas that we had and sequenced the large T locus, what we found were premature stop codons in each and every one of them. This was almost a signature mutation in integrated Merkel cell carcinoma virus. And what these polymorphisms did was to produce a tumor-specific truncated LT antigen. The small t was not affected due to the splicing pattern by these mutations. And when we looked at the expression of large T and small t, we could see, based on antibodies located at these epitopes that don't cross-react with each other, that both the small t and the large T are expressed in MCC tissues. Our control, which was an antibody against the SV40 large T, did not cross-react. And subsequent studies with the development of antibodies against the viral capsid protein and the ALTO, neither of those viral products were expressed. So in Merkel cell carcinoma tissues, only the large T and small t are expressed, and they are sufficient contributions of the virus to affect a transformed phenotype. So what we know now are the molecular steps in terms of MCV carcinogenesis to affect a tumor phenotype. We believe that that this is a ubiquitous infection, that almost everyone is infected and probably during childhood. But we believe that at some point subsequent to infection, there is reactivation. And that reactivation may be due to a transient immune suppression. It could be due to iatrogenic effects. But we feel that this is so because studies that we did on serologic assays, and this is with the collaboration and help and work we did with Chris Buck, also at the NIH, what we found was that the MCV serum titers for VP1 was really, really highly elevated in the MCV virus positive of MCC patients, but not in virus negative MCC patients, nor with blood donors on average. And so there is a problem here because I just showed you in the last slide that large T and small t are not expressed in the tumor cells. And yet these serologic assays would suggest that there was a spurt of infection where a lot of virus was produced. And what our model suggests, what I would like to suggest as a model is that with this reactivation, that there's this production of viral genome which is a messy process, that there is likely to be genomic integration of events into the human genome. Most of this would be stochastic, random integration, both on the viral genome as well as the host genome, and that there would be what would occur is that unless there were mutations, which I described previously that I told you about, there would be this unfettered replication, creating a lot of DNA damage, breaks, possibly collision of replication forks between the host and the virus, and eventually leading to the death of the cell. But if we had these truncation mutations that deleted the replicative functions of LT, what we would have would be the unfettered expression of the viral, quote, oncoproteins, the viral oncogenic expressions, clonal expansion of the host cell in which this happened. Two steps in which both are really rare events, hence the rarity of this cancer in the general population. So we continue to be interested in new pathogen discovery, and one of the diseases that we've always been intrigued by are post-transplant lymphoproliferative disorders. These are monoclonal or polyclonal proliferation that occur after solid organ or hematopoietic stem cell transplantation. And the majority of these are EBV positive, but there is a distinct subset of PTLDs that are EBV negative. And we hypothesized, or we were really intrigued by the fact that, that these were virus negative. And so Feng did DTS on 7 EBV negative PTLDs. And this was a collaboration that we had with Mike, Steve, and Ranga. These are hematopathologists at Pittsburgh. And the results from this analysis was negative. And so, you know, in thinking about it, we realized that DTS is dependent on first poly-A selection, which means that we are really biased towards linear transcripts. What if there was a circular RNA virus? Would we be able to find it? So Tuna Topton, a postdoc in our lab, decided to take this project on. And what she did was to, instead of poly-A select the RNA that was isolated from the PTLD, what she did was applied RNase R treatment, which is an exonuclease. What it would do would be to enrich for circular forms and deplete the linear RNA forms, make the cDNA library, do the sequencing, and look for and send the sequences through this DTS process. We didn't find a virus, but she did find circular forms, and these were circular RNA forms that that were enriched and that we could detect that derived from the viral genome. So let me just digress just a little bit in terms of circular RNAs. This field is becoming hot because it's now recognized that circular RNAs are another form of non-coding RNA in the cell that have very important functions. For their formation, usually the pre-RNA, in terms of what it does is that it makes use of the spliceosome mechanism that that is in the human cell. What the splicing does is that in the canonical splicing, what we would have would be the removal of the introns and the creation of sequential exons that are spliced together. There can be different patterns of splicing, but always it would be a sequential, you know, upstream exon followed by downstream exons. Well, in the formation of circular RNAs, what we have is an upstream donor or a downstream donor circularizing, going back to an upstream acceptor splice sequence. And what we would get in this case would be an exon that circularizes. This is another pattern where there's both forward splicing as well as backward splicing to make exonic only circular RNAs, but we can also have exon intron circular RNAs as well too. So many forms, but very, very interesting. And again, as I mentioned, something that people have become very much interested in. So what she found, again, were EBV circular RNAs. She found these at the BART locus. I'm going to truncate this a little because of the difficulties we had with the technical presentation. But at this locus, she found 2 backsplice junctions. But we really don't know unless we sequence these backsplice junctions and analyze them to see if they really are truly circular and whether they have that unique configuration of having a downstream site spliced to an upstream site. And as you see, we make use of what are called divergent primers. And from the geometry from this diagram, you can tell that if the divergent primers were to land on a linear DNA, you would not get a PCR product. However, if it landed on the circular forms, then you can go around the circle with PCR. And so she actually identified 4 different arrangements of these exons and introns in this BART region. Running out the PCR product, she showed that these circulars were found in EBV-associated diseases, specifically PTLD, but they have been found in other EBV-associated diseases as well as here she looked at 2 cell lines that were EBV infected. And so an interesting finding was that these circular RNAs had different cellular subcellular localizations, where the intron-containing circular BARTs were only found in the nucleus retained, whereas the exon intron circular or the exon introns were only found in the nucleus, whereas the exon only could be exported out to the cytoplasm. So in addition to the BARTs, there were a couple of other areas in the EBV genome which produced these circular RNAs, including the LMP region and the BHLF1 region. And these have been described by multiple groups. We said, why not? Let's look at KSHV, which is a closely related gamma herpes virus, as I told you. And we found 2 sites where KSHV elaborated circular RNAs. One is at the VIRF site, the viral interferon regulatory factor homolog 4. And then another site, which is really intriguing to us, is at a site, is the polyadenylated transcripts. And these are formed at really, really high copy numbers, hundreds of thousands of copy numbers per infected cell. And then K7.8 is exactly a non-coding transcript that goes in the opposite direction and overlaps with PAN. So this is a nice illustration. We used RNA in situ hybridization to find out some of the behaviors of these circular RNAs. And what you see is that this is using a technology called BaseScope. And so the linear LANA is a latency-associated protein that's constitutively expressed in cells that are infected with KSHV. And we can see that you have linear LANA. And then we designed probes that span the junction between the donor and the acceptor sites. And this is really the only way that you can tell the difference between a circular and a linear RNA. And we could find that in uninduced cells, which are latently infected with KSHV, this is a pattern that we see for the circular IRF4, PAN, and K7.3. But when we induce these cells so that the virus is undergoing lytic reactivation, some of the cells are dead, and there's very few after we treat and induce lytic replication in the cell line, but the LANA stays in the cells that can give a signal. Circular K7.3 is very similar to the uninduced state, but the circular PAN really, really increases, and that mirrors exactly what happens to the linear PAN transcripts. And interestingly, the circular VRF4 has no change. In latently infected cells, the circular VRF4 is expressed at high amounts, which doesn't change and doesn't become induced. And this is very different from the linear cognate of the circular form. This just shows that we were able to induce a lot of virus, take it through a sucrose gradient, treat it sequentially, plus or minus Triton 100 and micrococcal nuclease. And all this manipulation allows us to determine whether these circular RNAs are also packaged into virions. And we find evidence that these circular RNAs are indeed packaged in the virions here when we do the PCR. This is the circular VR4. And as I mentioned previously, the PAN and the 7.3, just really high copy number, backsplash junctions all over the place in this locus, nothing specific, but the production of a lot of circular RNAs. These are just controls of what we know usually come in with the virions. We also looked for circular RNAs in Merkel cell polyomavirus. Here is the genomic features as I showed you before. And here is where we mapped a circular RNA, the one that is the most abundance in Merkel cell carcinomas and in a infection model that we have for MCV. And this is work done by Bizu, Tuna, Hongzhou, Veronica, and Simon. So a lot of personnel were interested in what was happening here. And to make a long story short, what we found was that these MCV circular RNAs, you can see that here is the circular RNA. It overlaps with every T antigen transcript, but it also overlaps with a micro RNA that's produced by MCV. And the overlap in terms of sequence is 100%. And a lot of studies summarized here that says that there really is actually a regulatory function to these to the circular MCV RNA, and it's likely to regulate the expression of the large T antigen to, in the end, either inhibit MCV replication or increase MCV replication. So let me just end really quickly. Our lab is very excited about a proteomic approach to the discovery of tumor viruses. And it really takes advantage of virus biology. We know that there's an evolutionary pressure on viruses to reduce mRNA expression, but the viral proteins are really long-lived, all right? So this is to escape proteasomal degradation and presentation by MHC to activate CTL responses. And so Nathan Yates' group at the Biomedical Mass Spec Center here at Pittsburgh has been working on this quantitative differential mass spectroscopy to identify differences in peptides from human tissues that are subjected to different conditions. So we decided to look at 12 MCV positive and 11 MCV negative Merkel cell carcinomas as a pilot to see if truly can we detect viral peptides. And so this is a workflow. What we did was, first of all, made sure by immunohistochemistry that we had positive and negative MCV MCCs. After the samples, and what's great about this system is that you can use paraffin-embedded tissues, archival tissues. After the mass spec facility prepped them, they divided each one of these samples into 9 samples, and then which represented technical replicates in the system. They had machine replicates, a huge number of determinations. And what they found was that they were able to take out peptide features that were not previously identifiable as being human. And there were 498 or 500,000 of these. And when we looked at the top 20, and this was a laborious process for Pam and Shumei, because they had to hand sequence, they had to hand decipher each one of these peptides. And we found that the first, fourth, and the 14th and 15th peptides actually match back to MCV. And this is where they're located here. And you'll notice that from Merkel cell carcinomas, this makes sense because we're not producing the entire protein. We're only producing the truncated version, which only has the N-terminus intact. And here's just a dot plot that shows how specific in MCV positive versus MCV negative tumors these peptides occurred in. So in terms of a conclusion, I think that virus discovery, we have a lot of fun with it, but I think that there are things to be learned. There's really this interplay between immunity and carcinogenesis that we see over and over again. That's really a reflection of what is happening molecularly. We've learned that mutations in the normal flora can lead to carcinogenesis, not driver mutations in the human genome, but mutations in our normal viral flora. Through this process, sometimes we don't get what we want, but we have new science that comes out a lot when we pose the right questions. And we were able to find a new class of viral transcripts. And then I think that there's something to be said about really identifying an infectious cause to a human cancer, because then you have the hope of changing the landscape of human cancers, as can be seen with anti-cancer vaccines that have been developed, the examples being HPV and HBV. And then I'd like to really thank again the individuals that whose work that I presented. These are past alumni as well as present members of the lab, as well as these colleagues that I mentioned during my talk. And again, this was a really great pleasure for me to do this, a real treat. And thank you for your patience. And I'd be pleased to answer any questions you might have.
All right, thank you very much, Ewan, for the really spectacular talk. That was wonderful. So I wonder if I could start with just a very general question, which is, what is sort of your gut sense of how many cancers have undiscovered or underappreciated viral etiologies that have not yet been really discovered or reported?
So I'd like to just say that, we don't know everything right now, and we don't have a crystal ball. And I firmly feel that new technologies are coming online that will allow us to really think about this idea. I mean, we learned about the tie between immune suppression and virus-associated cancers. And it's been very fruitful using that as a guide to target us in terms of what diseases to look for. And there are other diseases that have an increased incidence when it is in the setting of immune suppression. But maybe we don't have the technology right now to look for something that may have a completely different pathogenesis. And that's very exciting to me. I can't give you a number, but I firmly believe that there are really more out there, and we just don't know enough about the process. And hopefully with each one that is discovered, we'll learn more about the science, we'll learn more about, how this carcinogenesis occurs.
Thank you. Okay, going to the chat box. Xue Wang has a question, a great talk. Epidemiology data show that HIV-infected individuals have a much higher risk of KS than transplant patients. And I wonder if you could comment on that. Is there a link between the two viruses, or what is your explanation for that?
The glib answer is that perhaps it's the degree of immune suppression. So we know that KS has really disappeared with heart therapy, right? So if you correct the immune suppression, you've got a cancer that completely disappears for the most part in patients with AIDS. So with transplant patients, we really can't take away their immune suppression because they'll lose their organ to see if the same thing happens. And then the degree of immune suppression with HIV, I think, plays a part in the differences between, it's the rate of occurrence of KS in iatrogenically suppressed individuals versus AIDS patients.
Question from Chris Buck. The Merkel cell polyomavirus genome codes a very large open reading frame just upstream of VP2, a location where other polyomaviruses encode the mysterious agnoprotein. Did any of the circular RNAs overlap with this particular open reading frame?
Yes, so I didn't put it in the slide for just simplicity and focus sake, but there are backsplice junctions that would suggest that there are circular RNAs in the late region as well.
Okay. From Joe Ziegelbauer, thank you for the presentation. Are you trying to use the proteomic approach to investigate EB negative PTLD cases?
We are, as we speak. Yes.
Excellent. Sounds like you can't say a whole lot about that yet.
Well, we don't have the analysis down yet.
What do you think of the possibility that novel viruses that are emerging, for example, SARS-CoV-2 or other emerging viruses, could cause cancer or be in some way associated with cancer?
I think it's possible. It's just anything's possible, I think.
Yep. Okay. Thomas Zhang, thanks for the excellent talk. Regarding this circular pan, if you have any idea how it's formed, this pan RNA region has no splicing in general.
Right, so that's really interesting. These pan transcripts, they have this interesting E&E motif that Joan Stites has postulated is necessary to replace the stability that comes about from splicing in genes. And so right now, we don't know why, we've seen in, for example, VRF4 that there are cryptic donor and acceptor sites that can create these circular RNAs that aren't used in forward splicing. But for the PANs, we just think it's really interesting, and we're very keen on following that locus up. No ideas right now.
Okay. So
you can let me know if you have any ideas, Thomas.
So I'm intrigued by these large T antigen truncation mutants with the Merkel cell polyomavirus. So what are the properties of those truncated T antigens relative to the full-length proteins? What do you think they're? I mean, I'm sure you know a lot about this. What they're actually doing, the truncation specifically.
Well, the obvious thing is that we don't have replication function at all. It's completely dead in the truncated one. There is a region in the very C terminus of the large T antigen protein. This is the wild type that's not truncated, that seems to have an effect on proliferation. And it hasn't been mapped, it hasn't been localized. And I just don't know at this point. We don't know. But in terms of transformation, let me think. I think that NASA really only did it with a truncated T antigen in terms of our lab and our studies. I don't know about other labs.
So it's hard to parse out simply the block to replication and potential transforming activity of these truncated large Ts.
Right. So with SV40, even though the mapping was not fine, it was a bipartite binding situation, and the P53 interaction was right in the middle of the OBD, the origin binding domain, as well as the helicase ATPase domain. So right now, even our full length MCV large T does not directly bind P53.
Okay, well, thank you very much. I don't see any further questions at the moment. So thank you again for the wonderful seminar. And we look forward to reading more about your exciting work in the literature in the years to come. Thank you very much.